Hello, welcome back. In my last video, we talked about the 5 I's of the sample processing in microbiology lab. If you haven't watched my previous video, the link is given above. Inoculation is the first step where we introduce the human sample into a medium that can allow the pathogen to multiply freely. The observable growth that appears on the medium is known as culture. Before we learn the techniques of inoculation, we must know various types of media that are used for inoculation. So, in this video, I will give you an overview of different types of culture media. Development of artificial media was the milestone achieved in late 1800s that gave a major stimulus to the rise of microbiology. For a successful culture, the artificial media must provide all growth requirements of a particular pathogen. The requirements of different bacteria vary from few inorganic compounds to a complex list of inorganic and organic compounds. That is why we have over 500 different types of media being used for culturing and identifying different types of microbes. Culture media can be kept in a test tube, flask, or petri dishes. You can compare culture media to the menu of a restaurant. The microbiologist has to prepare a specific meal of the choice for a specific pathogen. We can classify the media according to their physical state, chemical composition, and purpose. Based on their physical state, the media can be classified as liquid, semi-solid, or solid. The liquid media are water-based solutions that do not solidify at temperatures above freezing point and tend to flow freely when container is tilted. We use the term broth, milk, or infusion for them. They are made by dissolving various solutes in distilled water. Growth of microbes occurs throughout the liquid medium and can appear to be dispersed, cloudy, or particulate. Examples of liquid media include nutrient broth that contains beef extract and peptone in water. Coming to semi-solid medias, they do not flow freely and have a semi-solid consistency because they contain a small amount of solidifying agent, which can be either agar or gelatin, which thickens them. These media are used to determine the motility of bacteria and are also used for some biochemical reactions. Next is the solid medium. Solid media provides a firm solid surface on which bacteria can grow on top and form discrete colonies. They are used to isolate different types of bacteria growing on the solid medium from each other. We pick single colony and subculture it onto the fresh solid medium to obtain pure culture of the microbe for further identification. Liquifiable solid media are also called reversible solid media. They contain a solidifying agent that change their physical property in response to temperature. Most effective and widely used solidifying agent is agar. It is a complex polysaccharide isolated from algae Gelidium. Agar is solid at room temperature and liquefies at 100 degrees centigrade, and it remains liquid at 45 to 50 degrees Celsius, and therefore we can work with it easily at this temperature without getting burns, and it can be poured into the petri dishes at this temperature. It solidifies at 42 degrees centigrade. Another property of agar is that it can hold moisture and nutrients and can be used in the concentration ranging from 1% to 5%. Another solidifying agent is gelatin that is used in the concentrations of 10% to 15% to solidify the media. Non-liquefiable solid media are less versatile in their applications than agar media because they do not melt after once solidified. They include cooked meat media used for the cultivation of anaerobes. They start out liquid and get permanently coagulated and hardened by the moist heat after sterilization. Based on their chemical composition, the media are divided into synthetic or non-synthetic. Synthetic media or defined media are standardized and are reproducible. They contain pure organic and inorganic compounds in specific molecular content with an exact formula. If any one of the components is not chemically definable, the medium is called non-synthetic or complex medium. For example, the media containing blood, serum, meat extract infusions, yeast extract, peptone cannot have same exact molecular content and therefore are called non-synthetic media. So, nutrient broth, blood agar, though different in function and appearance, are all classified under complex non-synthetic media. Based on their purpose, we divide the media into general purpose media, enriched media, selective media, differential media, anaerobic media, specimen transport media, and enumeration media. The general purpose media allow wide variety of microbes to be grown in them. Examples are nutrient agar and brain heart infusion or BHI broth. Enriched media contain organic substances like blood, serum, hemoglobin, vitamins, and amino acids that allow the microbes with special requirements, also called the fastidious microbes, to grow in them. An example of enriched media is blood agar. It is made by adding sterile sheep, horse, or rabbit blood to the sterile agar. The media is used for the growth of pathogenic Neisseria species. Selective media are the ones that contain agents to inhibit the growth of common microbes and allow some specific microbes to grow on them. They are important in primary isolation from samples like feces, saliva, skin, water, and soil where we have large number of commensal bacteria. They speed up isolation by suppressing the unwanted background organisms and favoring the growth of desired ones. An example of selective media is mannitol salt agar. It contains high concentration of sodium chloride, that is 7.5%. Sodium chloride inhibits most of the human pathogens except Staphylococcus species that can be amplified from such mixed samples. Another example is bile salt containing media like McConkey agar and hectoin enteric agar that is used to isolate gram-negative rods, as most of the gram-positive bacteria are inhibited by bile salts. Other selective agents use dyes like methylene blue and crystal violet, and some use antimicrobial drugs and even acid. For example, selenite and brilliant green dyes are used to isolate salmonella from feces, and sodium azide is used to isolate enterococcal species from water and food. Next is differential media that allow multiple types of microbes to grow but are designed to display visible differences among them, either in the form of variation in their colony size or color on solid media, or in the formation of gas bubbles and precipitates in liquid media. These variations come from the presence of certain chemicals in these media and the different reaction of the microbes to them. McConkey agar is both selective as well as differential media. Reducing media contains substances to absorb oxygen, for example, thioglycolic acid or cysteine, to allow the growth of anaerobes. Thioglycollate in the broth uses all the oxygen in the liquid. The only source of oxygen for the bacteria is at the surface of the liquid. It differentiates between the aerobes, anaerobes, facultative anaerobes, microaerophiles, and aerotolerant organisms. Aerobes live only in the presence of oxygen, so they grow only near the surface. Anaerobes get killed in the oxygen and will grow only at the bottom. Facultative anaerobes grow throughout the medium, but maximum at the top because they generate more energy with oxygen. Microaerophiles need oxygen to survive but get poisoned by high oxygen, so they grow on upper part but below the top layer. Aerotolerant organisms grow throughout equally. Carbohydrate fermentation media contain sugars that can be converted to acid and gas, and a pH indicator shows this reaction. Transport media are used to maintain and preserve samples that cannot be transported to the lab immediately. They allow the delicate species that die rapidly to remain alive till they reach the labs. Transport media contain salts, buffers, and absorbents to prevent cell destruction by enzymes, pH changes, and toxic substances, but they do not support the growth of bacteria. Assay media are used to test the effectiveness of antimicrobial drugs. Enumeration media are used by industrial and environmental microbiologists to count the number of organisms in milk, water, food, soil, and other samples. So, this was an overview of the different types of media used in microbiology labs. Hope you liked the video. Do comment, like, subscribe, and share.